to talk about a very important topic, and that is gene expression. Because how does a gene go to a protein? And this is very important. So, we have an experiment called the beetle and the totem experiment with Neurospora crassa. And this is very low quality, so I will actually kind of skip the slide and go to the next one. And I'll open this in Campbell, but before I do so, I will talk about albino donkeys. So look at these donkeys. One of them, this is the brown or normal donkey. And then several centuries ago, there was a mutation. And a mutation is a change in the DNA. And it could be harmful, it could be helpful, or it could be neutral. And how does that one change in one base of DNA result in such a dramatic change in appearance? So first, before I reveal this, I will show this, what is called. This is a llama. See, this is a llama, and this has this, what is called brown pigment. And that brown pigment means it expresses a protein that helps you express that brown pigment. So the protein helps, and that protein is composed from a substance called DNA, which is produced into a substance called RNA, and then the RNA is produced into a substance called the protein, and then that protein, it's not really a protein, it's a polypeptide, so it'd be phosphorylated, or cleaved, or degraded, or like in this case, this function, it could be moved and its function remains, and then here you have this pigment, it is expressed, so the llama is brown. So it means that thing, it is an albino. It isn't like an albino, like llama. Okay, so the other donkeys that were here were actually albinos. They did not express the pigment, and that pigment expression could be very helpful. So how is this mutation affecting? So after all, there is a disease that is called albinism. And that disease called albinism can demonstrate the mutation. And then that donkey has albinism. And this mutation is slightly harmful because it places you at higher risk from skin cancer. But it is not a lethal disease. Homozygote recessive individuals, they sometimes are not sterile, and it is not lethal before reproductive age, so albinos could reproduce and create more albinos. So here we have this DNA, the gene for mutation, the mRNA, and then it becomes a protein. So here we have the beetle and the totem experiment, where we have the individual Neurospora cells that were placed on a complete medium, and they're subjected to X-rays to make mutations. And those are called mutagens. And each surviving cell, it formed a colony of genetically identical cells. And the cells from each colony were placed in a vial with only minimal medium. And the cells which did not grow were identified as nutritional mutants. And the cells from one nutritional mutant colony were placed in a series of vials that each contained minimal medium plus one additional nutrient. And here the cells grow when arginine is provided. And here we have this other one gene, one enzyme hypothesis. The experiment was built by Adrian SRB and Norman Horowitz. So we have the wild type that can grow on the minimal medium, ornithine, citrulline, and arginine. And class 1 can grow on this ornithine, the citrulline, and the arginine. and the class 2 can grow on citrulline or arginine, and class 3 requires arginine to grow. So the wild type can take the precursor and produce it into ornithine. It can, from the ornithine, produce citrulline. From the citrulline, it can produce arginine. So class 1 mutants cannot survive with the minimal medium because they cannot produce the precursor into the ornithine. But they can produce ornithine, so from ornithine they can produce citrulline, and from citrulline they can produce arginine. So in class 2 mutants, the precursor can be turned into ornithine, but the ornithine cannot be turned to citrulline, but the citrulline cannot be turned into arginine, the citrulline can be turned into arginine. In class 3 mutants, the precursor can be turned into ornithine, and the ornithine can be turned into citrulline, but the citrulline cannot be turned to arginine. The nutrient we need is arginine, so it requires arginine to grow. Here we have what is called the central dogma. So, oops, there is an error here. Okay, so back. We have DNA. So the general pathways is that DNA can turn into DNA, DNA can turn to RNA, RNA can turn to protein. And the very special pathways are that RNA can turn into DNA, and we'll describe this later in the slideshow. And RNA can replicate, but protein cannot. But RNA can turn to protein as a general mechanism. Here's how a gene turns into a protein. We have this promoter region, we have the TATA box, and we have the ATG right here. We have introns and exons, and then the transcription, the mRNA processing. We have the 5' cap. We have the stop codons, UGA, UAA, UAG, and then we have this 5 untranslated region with what is called a poly A tail, which is after that. But I have another document that I will show to you. I have already set it up. All right. And we have this DNA. Let's say it's TAA, so the RNA is UAA. We won't talk about that right now. So we have this 5' cap. And we have this little polyadenylation signal, which is AAUAAA, and that is in the 3' untranslated region. So it stimulates the addition of a poly A tail. And then we translate it. It begins with methionine, let's say lysine, serine, and let's say a bunch of amino acids like let's say phenylalanine, arginine, another one, and then let's say it's, another lysine, stop codon. And then we turn to active proteins, say it's sulfur, and there's some phosphorus, PO4. So first, RNA polymerase, together with one or more general transcription factors, binds to promoter DNA. And that RNA polymerase generates a transcription bubble which separates the two strands of the DNA helix. This is done by breaking the hydrogen bonds between complementary DNA nucleotides. RNA polymerase adds RNA nucleotides, which are complementary to the nucleotides of one DNA strand. RNA sugar phosphate backbone forms with assistance from RNA polymerase to form an RNA strand. Hydrogen bonds of the RNA-DNA helix break, freeing the newly synthesized RNA strand. If the cell has a nucleus, the RNA may be further processed. This may include polyadenylation, capping, and splicing. So, the important thing is that it could contain this 5' cap, which is a G, and a bunch of phosphates. Those 50 Ps, or what they call phosphates, after the guanine, which is a nitrogenous base found in DNA and RNA. And that poly A tail is 50 to 250 adenine nucleotides. Okay. And then the RNA may remain in the nucleus or exit to the cytoplasm through the nuclear pore complex. Okay, so you have a summary of eukaryotic transcription. So RNA polymerase adds complementary RNA nucleotides to that template DNA strand. And if the DNA is TAA, the RNA is UAA. But if we, let's say, place a G and a C and an A, there will still be a G and a C and an A. So the T is changed to U. Everything else is the same. And the various proteins again bind to this what is called a polyadenylation signal to a sequence AAUAAA near the 3' end of that pre-mRNA molecule. And about 10 to 30 nucleotides downstream, the cleavage and polyadenylation specificity factor, CPCF, it cuts the pre-messenger RNA. And the cap and the tail are added to the 5' and the 3' ends of the new RNA molecule as protection and to help it leave the nucleus. Now the 5 cap is a single G and like 50 to 250 Ps, or 50 Ps, and the 3' tail is about 250 adenines. So both ends are 3', which protects them from exonucleases, which target those 5' ends. And the non-coding introns are removed from the RNA strand, and the remaining coding sequences, or exons, are joined together. And then the 5' cap can then be recognized by what is called the nuclear pore complex, which allows the messenger RNA to leave the nucleus. Oops, so I accidentally flipped the slide again. So we have this data box, then a bunch of transcription factors, RNA polymerase 2, then it releases UTP, ATP, CTP, GTP, a bunch of phosphates, and then it begins transcription. Congratulations. Well, how does RNA processing go? So here we have the RNA polymerase that adds phosphates, then the splicing proteins come, and then the 3' end processing proteins. Okay, so just get the big picture. This is the DNA, and this is the end of transcription or translation, I don't remember, and then the beginning. Okay, so here we have what is called reverse transcription. We have this viral RNA, which turns into the target DNA, which could turn to viral DNA, which turns to viral RNA to produce new viruses. Okay, let's talk about spliceosomes. This is kind of poor quality again, so I will just flip the slide and go back to Campbell. There's a bunch of things here. We already covered them. Yeah, we did cover those. But just right before that, let me talk about transcription in prokaryotes. So first we have this initiation, where RNA polymerase binds the promoter, then wins the DNA, and then the polymerase moves downstream, and then they reform a double helix, and eventually the RNA transcript is released, and that polymerase detaches from the DNA. Here we've done that. Okay, so here we have what is called a spliceosome, where we have a pre-messenger RNA transcript with this pink intron and 2 red exons. And then the spliceosomes, they catalyze the cutting of that pre-messenger RNA and the splicing of the exons, which releases the intron for rapid degradation. Oops. So I don't remember where my window got gone. For some reason or another, I don't have my window open. Ah, there it is. There it is. Oops for the error. So we have this enzyme called aminoacyl transfer RNA that binds here. And then the small ribosomal subunit, it attaches itself to the 5' end of that messenger RNA strand. It moves along the messenger RNA until it finds a start codon in the first transfer RNA in the large ribosome subunit, they join it, and other transfer RNAs with anticodons matching the messenger RNA codons fall into place in the ribosome, and then the first transfer RNA drops off its amino acid and it breaks off and leaves to pick up another amino acid. And then when the ribosome reaches a stop codon, it releases the finished polypeptide. So here we have this, what is called the EPA site. This is explained in more detail in Campbell. Okay. This is like we have those 30S and 50S that bind in during the first stage and bind out during the second stage. So here we have initiation. We have the small subunit here, large subunit binds, and we have the EPA. And then here we have the or codon recognition, translocation, polypeptide bond formation, and then we repeat. And then the termination, we have the release factor right here, and then the free polypeptide, and then the whole thing gets released. Here we have what is called a transfer RNA. The second shape, like the secondary structure I meant, is like L-shaped. And this is described in Chapter 5 of Campbell, and I will complete this in a video which is called The Structure and Function of Large Biological Molecules. So after all, I wanted to inform you that I have another slide that you can view at the link that I will share a little bit later. It is a video from HHMI Biointeractive of how RNA folds. Okay, so here we have the next thing. When the nucleic acid is relieved of the constraint of being sort of lockstep with another strand in a double helix, it is free to take on all kinds of shapes because it can interact with other parts of itself. The structures that RNA can achieve have a wonderful variety, more on the order of what a polypeptide or protein can achieve, and this is what allows RNA to be a biocatalyst rather than just an informational molecule. Okay, here we have all the image sources. I'll just flip through. And this is not the end of the whole thing. I will just close this, and this is very important. And I have this, what is called mutations. But before we go to sickle cell and so on, I will show a very short animation. It is an animated GIF showing translation occurring. The EPA site showing the elongation. Okay, so done. Now I will move to what is called section. That's very important page, and we have multiple polypeptides. Before we go to mutation, here's the summary of transcription and translation in a eukaryotic cell. So RNA is transcribed from DNA, then it's spliced and modified, then it leaves the nucleus, and then the amino acid attaches to its proper transfer RNA with the help of a specific enzyme in ATP, and the succession of transfer RNAs add their amino acid to the polypeptide chain as the messenger RNA is moved through the ribosome, one codon at a time. When completed, the polypeptide is released from the ribosome. Okay, here we have what is called sickle cell disease. And this is a very important item, and there is an animation in the Pearson My Lab and Mastering study area. And it is a figure walkthrough. So here we have types of small-scale mutations that affect messenger RNA sequence. Let me get a little bit larger. Here we have the wild type where we have a methionine, a lysine, a phenylalanine, and a glycine. And then we have the nucleotide pair substitution, which could be, so we have an A instead of G and a T on the other strand. A U comes instead of C. And there's a codon table, and that codon table also exists in the figure walkthrough. This stands that this amino acid is a glycine, but this amino acid, because of redundancy in the genetic code, we have this glycine that remains a glycine. So nothing happens, so it's almost nothing. It could be modified on another level, like the chromatin level, but it usually will not affect the DNA or the messenger RNA or the protein. Okay, so missense can have a range of effects depending on the location within the proteins and the identity of the new amino acid. Here, thymine is changed instead of cytosine. Then an adenine gets changed, an adenine instead of guanine. Here we have a different amino acid, which we look on the codon table, is a serine. But how could we also read a codon table to identify how that amino acid change occurs? It is at the very beginning, not at the very beginning, but in chapter 17, in the very first section. So here, let's say we begin looking for an amino acid. First, its first letter, let's say, is a C. So first we go on the first letter. Let's say we're looking for CCA. So we have first letter C, like this. It could be any of these. Then go through the second mRNA base, which is C again. Then we look at the third mRNA base, which is A. So CCA is proline, or P. Okay. Nonsense. The effect depends on how close the new stop codon is to the beginning of the coding sequence. Here we have an adenine instead of thymine, a thymine. Then the uracil instead of adenine, and then it leads to a stop. UAG is stop. So what happens is that here the effect is very severe because the methionine is the only codon which gets released. Okay, nucleotide pair insertions or deletions. So here we insert an extra A, which leads to an extra U. This causes immediate nonsense. So it is both a frame shift and an immediate nonsense. This effect is very severe due to the nonsense mutation because you realize that the methionine gets released. No polypeptide is released. Here we miss, so we remove this A and we remove the U. Everything right before that amino acid, before that one nucleotide pair deletion, is the same. But what happens is that there's going to be an extensive missense. Here there are a lot of missense mutations whose effect is more severe than a single missense or even a single nonsense mutation, because there could be an extreme change in function and there could be a random stop. There's very likely to be a random stop codon. If there would not be a random stop codon in the sequence, then it would loop endlessly and it would spend too much ATP. We would actually stop it as a different process. The cell would run out of GTP. So the cell wouldn't be able to stop transcription, and if it does not produce more GTP, then it is going to again fail transcription. But the cell usually has a lot of GTP. So, a 3 nucleotide pair deletion does not cause an immediate stop or a missense. So what happens is that there is no frame shift because it is a multiple of 3. We have no more lysine, but everything else is okay. This is going to be okay if that amino acid is right next to. If you delete a stop codon, it is going to be very severe, especially unless that stop codon was not intended to be there and was due to a nonsense mutation. So these mutations are beneficial only when they eliminate the deleterious effects of another. So now we are no longer going to discuss all that difficult thing. It's very difficult, kind of. So now let's go to this transcript. This is about sickle cell disease, and it is available at the HHMI Biointeractive website by tapping in sickle cell anemia. So, when an individual, what happens is that the genes are lined up on chromosomes, and one has pairs of them with the exceptions of the sex chromosomes. And this means that you have two copies. The copies can be the same or they can be different. And if they're the same, they're called homozygous. And if they're called different, they're called heterozygous. So I have not created a video on this, but in the future, I will create a video called Mendel and the Gene Idea. This discusses the introduction to genetics and Mendel's work in detail, much more detail than this transcript does. So, when an individual finds a partner and reproduces, one of each pair of chromosomes is passed on. If the parents are both heterozygous, carrying one sickle cell and one normal gene, odds are 1 in 4 that the child will be sickle cell homozygous, 2 in 4 that the child will be heterozygous, and 1 in 4 that the child will carry 2 copies of the normal gene. In the absence of malaria, there is strong selection against the sickle cell gene. However, in a malarial environment, individuals born with 2 copies of the sickle cell gene and those born with 2 copies of the normal gene are both at a disadvantage. One gets sickle cell disease. The other is most vulnerable to malaria. Tony Allison's brilliant insight was that those carried just one sickle cell gene had an innate resistance to malaria. Malaria tipped the selective balance in favor of heterozygotes. The evolutionary trade-off is that protection from malaria comes at the cost of more sickle cell disease in the population. The sickle cell mutation was not the best genetic solution you might imagine to resist malaria. That's not how evolution works. It was the most available, a simple typo, A to T, and the gene that encodes hemoglobin. Mistakes are made in the copying of DNA in every generation. You and I were born with about 40 or 50 mutations that didn't exist in either of our parents. It's just part of the nature of copying 3 billion letters in the process of reproduction. And when those mistakes arise, a typo arises in the globin genes. For most of us, it would be a bad thing. But if you live in a malarial area, it gives you an edge against the malarial parasite. So that mutation is retained. This malarial mutation is a missense mutation. And that missense mutation is really severe in some cases, so it is both severe and beneficial. And this is how the process of evolution and natural selection works. This is a real, human example of how that works. Thank you.